Hello everyone, welcome to Pathology. We'll get started in the next 3 minutes. Thank you for coming. Good afternoon, everybody. Thank you for coming to my morning show. We'll get started in the next 2 minutes. Thank you, everyone. Thank you. Can everybody hear me? Can you all hear me?
I can.
Okay, that's good.
Yeah.
I think today is very special. Excuse me. Today is very special presentation, and so the topic is dedifferentiation and redifferentiation, tissue culture engineering of articular cartilage. I usually start this with a quote, and this quote I will translate it in English, but this is an Igbo quote. This topic is very, very important because it deals with osteoporosis and arthritis. And in Igbo culture, there's a adage that we use. It's ugu mota mbaji ukwu agadi nwanyị amụta agakata akwụlụ. It means the heel is inclined to cause severe joint injuries to elderly women. As such, elderly women have learned to climb the hill with patience and resilience. Right, so there might be some type of epidemiological connection to this word, but also there's another one. This man is well known in my tribe, and Udembweke is a herbalist, and he usually, when he get invited to town hall meetings, those days about over 200 years ago. Mostly people don't have cars. People have to go from their villages and walk to the town hall meeting to participate in the meeting. He usually start by saying, Ndo aka na ebu, na ndo akpa na ebu, ndono. That means that for those with legs and hands pain or arthritis, you have my sympathy. So arthritis and osteoporosis is not something new, it's something generational issue. So that being said, there are certain things I would also like to discuss. But before that, actually, let me do that before I introduce the presenters. So one of the important things with arthritis, I'm going to talk about the global aspect of arthritis. The knee is the most frequently affected joint pain, followed by the hip and the hands. In 2019, approximately 528 million people globally were living with osteoarthritis, an increase of 113% since 1990. Roughly 73% of people living with osteoarthritis are older than 55 years old, and 60% are female. The prevalence of arthritis globally is about 365 million. The knee is the most frequently affected joint, followed by hip and hands. More than 70% of women and 35% of men with osteoarthritis of the knee are overweight. So now it connects to the biophysics of the body. About 344 million people living with osteoarthritis expressing moderate or severe pain could benefit from rehabilitations. With aging population and increasing rates of obesity and injury, the prevalence of osteoarthritis is expected to continue to increase globally. Osteoarthritis is not an avoidable consequence of age. This is World Health Organization. Some of the causes and risk factors associated with this problem, there are several of them, but I will just mention very few. Injury to the joint, fracture, strain, repeated stress in sports or at work, pre-existing joint diseases such as rheumatoid arthritis or gout. Specific metabolic diseases such as diabetes can also be linked to this. Obesity is one of the biggest ones, specifically for hip and knee arthritis, as characterized by metabolic abnormalities, systemic inflammation, and contributing to excessive load on the joint. So if we look at obesity, for example, or generally overweight, about 1.9 billion people are overweight, with over 650 million people of those that are overweight that are obese. And now this is globally. Also, another thing is that arthritis can be genetically linked. There's also socio-demographic factors, age, gender, or sex. We already seen that among all those that have this condition, 60% are women or female. This is important because we need to understand the risk factors associated with this globally and take appropriate remedies to mitigate this issue. We can reduce overuse of the joint, developing best work practices if the problem is related to work and just personal lifestyle. Promoting healthy lifestyle is also very important. Regular physical activity, maintaining a normal body weight is very, very important. Now we know that we're just coming out of COVID-19, but we are talking about a condition where over 500 million people, prevalence-wise, people are currently living with this. So it's a big issue that we need to address. This being said, let me introduce the presenters that I have here. First, I'll start with Professor Dr. Marc Facciotti. I think if I start reading everything about Professor Marc, that will take the whole show to read. He's a well-accomplished professor at UC Davis, BS in biochemistry, PhD in biophysics, postdoctoral also in biophysics, and postdoctoral in systems biology. Professor Marc Facciotti received a BS in biochemistry from UC Davis in 1997. He trained in structural biology with Professor Robert Glacier at UC Berkeley and completed a postdoctoral fellowship with Laurie Hood at Institute for System Biology. Facciotti's research background and academic training is diverse, including a broad background in biochemistry, molecular biophysics, structural biology, microbiology, systems, and synthetic biology. He has been a faculty member at the University of California, Davis since 2008, where he has studied gene regulation in a diversity of microbial and eukaryotic systems and worked on the development of educational technologies and creating space that expand access to research experiences for undergrad students. So, Professor Marc, you're really welcome. I'm excited to hear what you have to discuss in this topic area and inform us more so that we can learn about this research you've been doing. The co-presenter here is no one other than Dr. Facciotti. Dr. Facciotti is actually one of the pioneers of biotechnology in the United States and around the world. He's well-versed in plant physiology and plant biotechnology. This man has done a lot of work in biotechnology. So again, if I read a lot of stuff that Dr. Facciotti has done, we would take all day to read. Dr. Facciotti, Daniel Facciotti, specialized in cell differentiation and plant regeneration, effects of ploidy on cell physiology. He studied in a lot of stuff in, I think it's soybean, as applied mainly to genetic engineering on herbicide resistance, oil composition, and puffer oils. I mean, he can talk about maybe later on what this puffer means. But regulation of science as applied for commercialization of genetically engineered plants and oil-derived products. Mutagenesis technology as applied to the generation of tailing, which means targeted induced local lesions in genomes. Mutation libraries and selection of point mutation in genes affecting herbicide resistant fatty acid metabolism and food/feed quality. Cell now currently doing cell dedifferentiation and redifferentiation as applied to tissue engineering of articulate cartilage chondrocytes. So without further ado, welcome to Dr. Professor Marc Facciotti and Dr. Daniel Facciotti. You're welcome. So I turn it over to you guys to, say a few words and let's kick off.
Well, thank you, Ndukako. Thanks for the invitation to present. And are you seeing, just to make sure before we get started, are you seeing the shared presentation on the screen?
Yes, we're
okay,
seeing.
Perfect. So thank you, as I mentioned, for the invitation here, and also thank you to the folks in the audience who are taking some of their time to hear about the work that we've been doing. This is a particularly special project for me because I get to work on it with my father, Daniel Facciotti, and so we've finally gotten around to doing something together in science, and this is some of the product here. So it's great to be able to share that with everyone. There's obviously more people that have been involved in this project, and I just want to make sure to recognize them all. Hansu Kim was a PhD student with me. She graduated last year and is off to better things. Andrew Yao was scheduled to co-present with us. He's an RA in the lab, but he got taken away from us today due to some family responsibilities, so he sends his apologies. And Haven and Rachel were undergraduate students that worked with us on establishing this model system. I also want to give some thanks to a faculty colleague of mine, Soichiro Yamada, who's contributed some of his equipment for us, and also to Department of Biomedical Engineering who provided some of the funding for the work. So without further ado here, I'm going to kick it over to Daniel, and he'll start us off with some of the background and motivation for the work.
Oh, so before Daniel, you start. So
yeah,
here we will start with Daniel discussing the problem, background, purpose, and significance, and
yes.
the research question they're addressing in this experimental study. So I'll turn it over to you, Daniel. Go ahead.
Okay, so one thing that you forgot to mention in your introduction, thanks for, again, the invitation, but you forgot to say that we work together.
Oh, yeah. So now let me say that, you know, Dr.
On engineering canola.
Right. Dr. Facciotti, Daniel Facciotti, since there's two Facciottis here, one is a dad and one is a son, right? Dr. Facciotti, Daniel Facciotti is one of my key mentors when I started in working in biotechnology industry. And one thing actually that triggers me to be more inclined with Daniel's approach is that among all the research scientists, principal scientists we have at the time in the biotechnology industry, he's the only one that's really interested in actually doing real work and research. So I always tell him that and say, among all the so-called PhD at the time doing this work, you're the one coming in the lab actually so interested in asking questions and experimenting on things. And I work with him a lot with a lot of stuff. You know, we change protocol, we manipulate things to see what we see. And in fact, to be honest, most of our private discussion we have was actually correct. But I'm not going to go into that. But, you know, when you work in organization, there are certain things you will see that doesn't make sense. But obviously, you have to do the work. But, you know, that's for a different story.
Yeah, and Dukaku was one of the most entrepreneurial guys I've worked with. So it was fun.
Yeah,
it was very much fun. All right, so let's start right now. I will try to put this study within the context of tissue engineering and regenerative medicine. So clearly, regenerative medicine and tissue engineering are as old, if I can say, as humanity. The goal is clearly to fix what's broken in a body, or repair, substitute, like I put here. Next. It's not only for fun or for scientific fun that you do that, but there is a real need in the United States. I put here, there is about 100,000 people who are waiting for organs and only 14,000 deceased donors. So you can see that there is really a lot of needs for engineering tissues and regenerative medicine. So besides this need, clearly regenerative medicine can be used for studying. If you have a little organ, then you can study not only the function of these organs, but the diseases in this organ, and also how to treatment. For example, for pharmaceutical use, it can be really an interesting thing to have. Just in passing, biomaterials, you can practically include everything you want in biomaterials. It can be synthetic biomaterial for as long as it's biocompatible. Tissue transplants are included in regenerative medicine. Cells or cells combination, cells and any kind of biomaterial, for as long as they are going to supply the material to fix an injured tissue or an injured organ. Okay, a few examples of what has been done. You probably heard in the press a little bit of everything. So tissues and organs that have been generated, liver cells, pancreatic cells, in fact, for the pancreas, there are little organoids that have been also transferred into animals, and they produce insulin. So the future, perhaps, creating pancreatic cells that can be implanted in people. For the moment, I think most of what you can see here in this slide is being applied to animals. And it's still at the state of the research, advanced research, but still research. For two types of tissues, skin and cartilage, there are, however, a few products that exist that have been already approved by FDA, and some are on the market, particularly one in cartilage. Next. So since we talk about cartilage, here is cartilage, or at least you can recognize here a knee joint laterally and seen from the front. So with the femur, the tibia, the patella that is visible, and the articular cartilage that we are talking about, it is the tissue that covers the epiphysis of the bones, generally speaking, of the long bones that are in the articulations, the knee, the shoulder, the elbow, the hips. They are all covered with this articular cartilage. So this articular cartilage, also called hyaline cartilage because of its appearance, it's kind of glossy and translucent. So you can see on the right, and I see my picture on the right. I would like to take it off. Okay, you can see that there is a meniscus. There is the articular cartilage, and underneath there is a meniscus. Meniscus is also a type of cartilage, but not the same. It's called fibrocartilage, and the big difference is that it contains a lot of collagen 1, not only collagen 2, like the articular cartilage, hyaline cartilage. So hyaline cartilage, it's only 2 to 4 millimeter thick. It is not irrigated and not innervated. I'm going to give you a few characteristics. It is the record for low cell density in a tissue. As you can see, the little dark spots that you can see in these slides, maybe Marc, you can show them. Okay, those are the cells. They are the chondrocytes, the cells of the articular cartilage. All the rest of it is extracellular matrix that is produced by these cells. So there are very few cells in this tissue. So 5%, low density, extracellular matrix, 95%. Composition, and just remember that it is on a 2 to 4 millimeter thick. So next. All right, and the composition, you can see that it is 72% water, only 5% cells, and 25% macromolecules. These macromolecules can be subdivided in two. Proteoglycans are about half, 25% are proteins, non-collagenous proteins, and 25% approximately are collagen type 2. And one of the characteristics of this tissue is collagen type 2. So about the proteoglycans, you have here an example. You probably recognize material, at least pills that you can find at the store, chondroitin sulfate, keratin sulfate, hyaluronic acid. You probably can find that at the store, or sometimes you have injection at the doctor of hyaluronic acid. What do these molecules do? They keep water in it, and they attach to water, they keep it into the articulation, and they produce really something that is a buffer for the knee or for the other articulation. So the next, here, you can see that it's not a uniform tissue. 4 zones can be distinguished in this tissue. There is a superficial zone that has a certain pattern of cell dispositions. And then there are 3 more zones. The last zones at the bottom is calcified, and that allows the tissue to be connected to the bone itself. Otherwise, it would slide off. These cells, not only they have a different phenotype, or perhaps what we could say a differentiation state, they are not the same, and they also have different metabolisms, which causes perhaps some questions later on, or will give us some questions later on the choice of cells that we're going to use. On these 2 to 4 millimeters, if you look now at the direction that the collagen 2 fibers have, you can see that it's a remarkable structure. So on the top, all the collagen is parallel to the surface, and that allows the knee to slide frictionless on each other, the bones on the knee to slide on each other. Underneath, however, you can see that there are structures that are like arches. And I would say, I always take the example, arches in a cathedral. They support the roof, but they also distribute all the weight underneath. And in this particular case, clearly they distribute the weight on the bones that are under the cartilage. So next, sorry, Marc, can put it back, yeah. So all this tissue here is not formed at birth. At birth, it is immature. That means that it doesn't have this structure at all. It matures under the influence of weight. I think as you grow, you put more weight on it, and this weight is going to push these cells to differentiate in the way you see it here. And it takes quite a long time to mature. So one thing is that you can immediately think, if you try to redo this work, right, to regenerate this tissue that it took a long time to be formed, it's going to be extremely hard. One more thing to say is that this tissue is really unable to heal itself. It's not like skin or a bone. Skin, you know that you're going to have a scab, and that's pretty much it. The bone is going to reform, is going to fix itself. It will take perhaps a few weeks. The cartilage is not, at least not when it is mature. It is reported, at least by experiments on animals, that young animals where the cartilage is immature, you can wound the cartilage and it will fix itself, but not in mature cartilage. That doesn't happen. Next. So we talked about arthritis and osteoarthritis first. What can damage this tissue, this very thin tissue? Well, accidental trauma, we have seen that, but essentially it is arthritis, and osteoarthritis is the main one. I listed here a few of the other forms of arthritis that exist. All of them end up always by a loss of tissue, a loss of cells. It is a degenerative disease and always end up with loss of tissues. And as I said, this tissue doesn't heal itself. So long requirement. Here, maybe something that Dukaku has mentioned earlier, I also had it here. In the United States, I think the cost of osteoarthritis is estimated to be at about $300 billion a year. We were talking about what are the knee and the hip is where the disease is more prevalent. And there are every year about 850,000 knee and hip replacement, and it's projected to go up to 2 million by 2030. And here a few estimates in the United States by the CDC. And as you can see, the numbers that you can see for the world population, as Andukanko has mentioned, are not negligibles. In fact, they are significant numbers. So it is a disease that requires some more attention, in my opinion. Next. Okay, talking about the causes, and here I will essentially, since I myself suffer of osteoarthritis in one of the knees that is going to be operated, I have my own conception of how it works. It's an inflammatory vicious cycle that occurs. It starts perhaps with a trauma, an accident, sport injuries in my case. In other people, it can be wear and tear. If you have a misalignment in the articulation, if you are too heavy, then you can have some wear and tear, and you can overuse your joints. And then I put also the age. It is true that this happens to aging people more often to the young people, but I don't think that age is a cause of it, because there are a lot of old people that don't have osteoarthritis. It's not obligatory, but I think it can be combined with other aspects of aging. In fact, while working on this project, I looked quite often about the research that is done on senescence, which is very interesting when you reach a certain age. But age, at least, can affect osteoarthritis because cells don't act as quickly. They don't, for example, excrete as rapidly as younger cells. And so it can accentuate. But again, it's not because you're aging that you're going to have necessarily osteoarthritis, especially if you didn't have a trauma or if there is no wear and tear. I put here on the right one of these cycling, if you want, vicious cycling. What I call vicious is a positive feedback. That means you start with a cell and that goes around. So alarmings are received by macrophages that produce pro-inflammatory compounds on fibroblasts. Fibroblasts, we don't know exactly how they affect the chondrocyte, but in the end, you have these metalloproteins that are destroying the MMP3, destroyed collagen 2, for example. And then you have the result of the destructions on the extracellular matrix. They go back to the osteoclast and everything starts all over again. So you have this positive feedback. Okay, next. This is the result. You can have on the left side, you can see femur and tibia. They are practically, everything has disappeared between the two, and the two bones are not frictionless sliding over each other, but they really encroach on each other. And since the bone now that is uncovered is innervated, it will hurt you quite a lot. So that's what hurts. The cartilage doesn't have any innervation, so it won't hurt, but the bones that is underneath will. On the right side, you can see here some quite large damage to the cartilage, which is white, and underneath in pink, you have the bone. It's pink because it's irrigated by blood. Very often there are asperities. The bones start growing on that, and it forms asperities that then friction with the other bone, and that causes pain too. So when you have arthritis, osteoarthritis, it's painful. That's the first thing that you feel. Next. So what are the therapies, or how do you mitigate pain or alleviate the disease? So you have pharmaceuticals, all the anti-inflammatory analgesics that you have, ibuprofen and others, you will find them. Antioxidants too, to slow down the progression of the disease. And then xenomorphics and senolytics. Those are still mainly under research or only given by doctors. They have to be prescribed. You won't find them over the counter. Xenomorphic help the cells, old cells, excrete a certain number of compounds that may affect other cells. The senolytics, as for them, they target old cells and they kill them. And it was noticed that if in a tissue you kill old cells, the other cells do much better. The cells that are still healthy do much better. So, but that's still, I think, a research product. You can also do surgery. That's probably still largely used. Realignment and distraction. So realignment is really realigning the joints, and that's quite a big operation. Distraction, you have an example here of distraction, where you keep the components of the joint separated from each other. And you can see with this contraction here that goes into the bones, screws, and everything, and it keeps, it liberates a little bit, keeps free the cartilage so that it can regenerate. This is done only in young people. It works only in young people. Debridement and washing, as I said, if you have asperities and they break down due to friction, they cause additional, not only pain, but additional inflammation. So you go into the knee and you wash them off, and that helps. Microfracture was quite popular a few years back, and it consists in drilling up to the bone. You drill the cartilage up to the bone in the areas that are wounded or where there is a defect in the cartilage. And you do that, you go into the bone because a certain number of mesenchymal cells come out and try to repair. It has been abandoned. It doesn't work that well for large defects in the cartilage. Then you have osteochondral plugs that are little portion. You go into an area of the hyaline cartilage that does not carry any weight, so laterally, and you take little plugs, and then you use those plugs to fix the areas that are damaged. This one, I have to say, it's quite successful. It's probably one of the most successful systems that have been devised in areas. If the wound, however, it's quite small, so less than 2 square centimeters. Less than 2 square centimeters, because even if you take the cartilage, plugs of cartilage in healthy areas that don't carry weight, you can only do it in a limited way. You cannot just, because otherwise you're going to damage too much the knee. Okay, next. Finally, what we are interested in is fixing this damaged cartilage with cells. Now here I put a certain number of cells, stem cells. They are quite popular. Embryo stem cells, mesenchymal stem cells, induce stem cells, for example, from fat cells. You can do that. However, all these cells are very difficult to recruit, very difficult to obtain. So most likely you're going to have what is called here the ACI, which is the autologous chondrocyte implantation. And that can happen with growing these cells in with or without a scaffold. So what do you do here? You have to take a few cells in the cartilage, again, in areas of the cartilage that are not carrying weight, and then you're going to grow them in culture, and you re-inject them in the knee. I think, can you go back one? Yeah. So, and then the C portion that you have here, it's again for the future, is not really much done at the moment, but it is matrix assisted chondrocyte implantation. And you can see that you can mix them with mesenchymal cells, or even you can think about putting together chondrocytes plus genetically engineered cells that can guide perhaps that cells to differentiate properly. Okay, next. So this is how it's done. You can recruit cells or enzymatically isolate from tissue. I think most of the time you're going to take a portion of the cartilage, healthy cartilage, and you're going to digest it with collagenases and other proteases and obtain cells. Then you expand the cells in culture. That means you multiply. You start with 10 cells and you're going to go up to 10,000 or 20,000 or more. And you can do that in just freely, or you can introduce the cells in a scaffold. And perhaps it's too long to really explain with the scaffold, but it is something that will probably give you a shape that is more desirable than the cells. Then you implant again directly the cells in the lesions, or you can implant them after you have followed a different differentiation. We'll see why in a moment. Most steps that are done here or that have been done so far are carried out empirically, and it is by trial and error. So next, in all the cases that you have seen, you have the formation of fibrocartilage and not hyaline cartilage. Does that provide relief? Yes, certainly it does, at least according to most of the people who are asked for. But the cartilage that is produced by all these techniques, it's not as good as the native cartilage, and it doesn't last as long. So you can ask that to yourself, did I use the wrong cells as inoculum? In other words, the cells were not sufficiently differentiated. Differentiated meaning they were not looking, or they were not differentiated in the way the original cells were, the native cells were. Or is it, even if I introduce cells in a sick articular environment, the cells are good at the beginning, but if they are in a sick articular environment, they're not going to last, and they're going to switch or differentiate into something different. Or perhaps both, you start with the wrong cells and so on. Next, and I think that will be my last one. So if you think a little bit, you start thinking about cell sources, and we talk about differentiation state and ability to dedifferentiate and redifferentiate. And so I can just say, differentiate, a cell of a skin is differentiated in a different way than a cell of a bone, right? They took different paths into their differentiation. Dedifferentiate is just the ability to, again, go backwards and being able to divide again. And that's what you need when you want to expand cells. You start with a few cells, and then you want a lot of cells, because sometimes the damages are large. But if they're able to divide and they differentiate, you don't want that to continue forever. So you need to be able to redifferentiate them. In other words, go back to their history and redifferentiate them as articular cartilage cells. So now, when we say all that, dedifferentiate, differentiate, redifferentiate, I think, what are we really talking about? And when you try to understand what you're really talking about, at least for me, I was unable to say, oh yeah, I understand, I know. No, I don't know. Is it phenotypically? Or is it because of certain metabolic activities? Or in terms of gene expression? Or in terms, I think that's another important part of future research, I believe, which will be in terms of epigenetic state. So couldn't understand that. And I think I asked around, and yeah, there is some knowledge about the differentiation, but it's not very advanced. So, and this is in part, I think Marc will talk about more about our approach, but what we try to accomplish is to have answering this question, but in a scientific way, to put down scientific foundations. And initially, it will be based on gene expression. And now I let Marc take over.
Yeah, thank you so much, Dr. Daniel Facciotti. This is very interesting. And for me, what I got from this, the differentiation is going back in time. And redifferentiation, you trying to separate cells to form new things. So this is very, very important in this 21st century experimental design research in futuristic moment of renaissance in knowledge, right? So this is very, very good. And I like the fact that you clearly explain what University of California, Davis is trying to do, specifically Marc's lab is trying to do, so that our audience, mostly who are epidemiologists or in public health, even some nursing practitioners are in the house here listening to what you are presenting today. Some of them are now, molecularly tailored, right? So it's really, really interested to just get the overall picture, which Dr. Daniel Facciotti has explained. So now, Professor Marc, take us step by step.
All right.
To get into the methodology and findings
Sure.
and everything, all the lessons learned so far. But I had to warn the audience, this is purely an experimental research. So I want to make that clear.
So I'll see what I can get done with the time that remains here. So I want to give credit to a colleague of ours, Professor Athanasio, who's now at UC Irvine, who really launched us into this work. Professor Athanasio was a mechanical engineer, but he got into biology and really has been working for many years on the problem of engineering new cartilage. And you can see some of the material that they generate in their lab on the right. These little discs are little discs of cartilage that were grown in a method that Danielle already described. And Kyriakos was one of the early pioneers involved in the autograft technologies that Danielle described earlier as well. And he's moved on to trying to figure out how to tissue engineer new cartilage as kind of a next phase. And so he's part of the inspiration for some of this work. There's another bit of inspiration here, and this might be viewed as kind of a framework for our thinking, if you want. Is to think of cells as little biocomputers. Here I have a cell cartoon represented, and the key thing is that the cells have all sorts of sensors that are arrayed within and across a cell that are harvesting information all the time about what's happening in their environment and feeding that information into the cell, into the nucleus, and maybe even not to the nucleus, but to other parts in order to respond to all those stimuli that they're receiving. And they're receiving information from hundreds of different sources all at once and somehow processing that. So that's part of our kind of a foundational view here. And some of these signals that go to the nucleus, if we take the arrow as saying, let's peek into the nucleus here, and we zoom in on what's happening at the nucleus, we know that the nucleus holds all the DNA for the cell and that the DNA has a bunch of these genes that are encoded on it. And those genes are expressed through the activity of proteins called transcription factors that bind near promoters and generate mRNA. That's the first component of gene expression. And so you may have two things that happen. One is you have a production of RNA, or you may turn off a gene. So you can have control depending on what you're sensing in the environment to either turn on genes or turn off genes. So this is one of the primary responses of cells in every tissue and every organism for responding to the environment. And we posit that patterns of gene expression, so if we look across all the genes and we look at what's happening up or down in those are patterns of expression, that can be a high-resolution fingerprint of cell state. So it tells us something about what state the cell is in. So that's also a piece of background here. Now, cartilage, as Danielle already outlined, the growth of cartilage in vivo is a process of cells starting as stem cells, here mesenchymal stem cells, and slowly sensing changes in their environment and triggering different genetic programs so that they mature ultimately into mature chondrocytes with a rich extracellular matrix that's supporting the joint. So this is the development process that we would like to replicate to make new cartilage to help people with osteoarthritis or acute injury, needs to somehow mimic, or we think of it as maybe mimicking this natural process of development where certain genes are being turned on and off over time. Now, that process happens over time, I just said, but we also need to think, and we'll bring back this idea that the tissue is not homogeneous, it's heterogeneous in its depth, as Danielle mentioned earlier. And so there are patterns of gene expression that are not the same in cells at the top versus cells in the middle and cells at the bottom. And so one of our goals ultimately is to understand how is it that we can program cells to behave properly if they're going to be cells that are on the top of the material, cells that are going to be in the middle of the material, how do we help program those and direct those genetic programs and those near the bottom of the material. So, that's some of the physical background. Now, tissue engineers approach this in a very simplistic way. The basic premise of tissue engineering is that cells are smart, and if you give them the proper signals at the right time and at the right abundance, that they'll figure out how to make it happen. And so what do tissue engineers do? Well, they go on a hunt. They take some cells, they expand them, and then they start giving them different stimuli, growth factors, temperature, pressure, all sorts of signals that the cells might receive. And they try to mimic what happens in living systems. And they do this over and over again. An assay, right? They'll start with cells, different types of cells, as Danielle mentioned, different types of scaffolds, different types of signals, and they grow the tissue, and then they test the tissue to see if it's something like the native tissue. So this is the main approach to tissue engineering. It's, as Danielle mentioned, a bit ad hoc and a search through condition space, I call it, hoping that you find the right combination of conditions so that the cells on their own figure out what to do. Now, the problem with this is that for the most part, Danielle also mentioned, most of the time you end up with, in the terms of cartilage, fibrocartilage and not hyaline cartilage. And you end up with tissues and materials that don't have the stratification. They're homogeneous materials. They're not stratified like the real native cartilage. And so you go through all this process, 20 years of this, and you end up with tissues that don't work for the most part, or they don't last. How about that? And so you can ask yourself, why is that? And what can we contribute to this story? And one of the things we noticed is that very little work has been done trying to understand what happens in the process of making these new tissues. Most of the time, if you're lucky and you look at papers of people that are doing this work, they're trying to characterize, if they characterize the process even, they are looking at maybe 5 to 10 markers, cellular markers of that are supposedly informative of what might be happening. So we thought, well, that's a small amount of information for the huge amount of genetic reprogramming that's happening during development. So maybe we can do something there to contribute. And then the second kind of thing this strikes us is that there are very few models of the regulatory structures that control development in most of these tissues, and they're hardly ever, if ever, used in the process of tissue engineering. And I'll say just a little bit more about why those are important through analogy here. So here's a radio. It's a device that takes environmental information, in this case, electromagnetic waves. Somehow in this box, we don't have to know how to wear it. We just flip it on and it works. But we know that in this box, that box is capable of taking those electromagnetic waves, those environmental signals, and converting them to sound. In biology, we can say, well, the cell is similar. it takes all this environmental information and it spits out a phenotype, some behavior, some property of the cell. The phenotype is what we're trying to engineer in these cells. We want them to be nice articular chondrocytes. So let's come back to the radio. How do we understand how to build and modify the properties of a radio? Well, if we're an electrical engineer, what do? We go in and we have a parts list. We know if we were to break open the radio that we would find, this is an old radio, by the way, right? All these old electronic parts, but for the analogy, it works. You'll have all these little parts in there that contribute to the function of the radio. But what else do you need? You need to know how those parts are connected to one another. You can't just throw a bunch of parts together in a box and hope to get a radio. You need to connect them in very specific ways. And if you know how the rules for connecting this radio and the parts therein, you can also modify the behavior of that radio by adding new parts or changing the connections, so forth. And so that's kind of the idea that we're going after. This is a model of how the radio works that can be used to drive rational experimentation and engineering of the material, or in this case, the radio, to a desired phenotype or behavior. So in biology, the parts are the genome sequence. We're getting very good at sequencing genomes. I think everybody's comfortable with that. And the components of the wiring diagram for the cell are really composed of these transcription factors and the connections that those transcription factors have to turning on and off genes. We can represent those in kind of a cartoon format. Here's a transcription factor, and this graph says this transcription factor is connected to this gene, and it controls the behavior of that gene. Of course, there's more than 2 genes in the genome, right? We can grow this out into a little bit more complex network and even more complex network. And that's really what's inside the cell that we need to understand if we want to drive different phenotypes. Now, is there any basis for this being a promising approach to thinking about tissue engineering and development? And I can go back to some old work done in the early 2000s by Eric Davidson and sea urchin. Here they've elucidated a regulatory network. This is basically an expanded version of the figure I showed you before, a regulatory network for the development of sea urchin. And what they found was when you look at parts of this regulatory network, in dark lines here are genes that are active, and in grayed outlines, there's parts of the regulatory network that are not active. So here at 6 hours, you can see one part of the regulatory network is active. At 12 hours, information is spreading through this regulatory network and other parts are lighting up. At 18 hours, information is spreading throughout other parts of the regulatory network, turning on programs of gene expression that guide the development of early development in sea urchin. It turns out that this type of network was sufficient for Eric Davidson to then go and change some of the connections in this network in a predictable way that changed the development of the sea urchin. And so they were able to use this kind of model to direct the development of sea urchin, in this case, the formation of the gut. And so we hope to apply a similar approach to understanding the development of cartilage. All right, so what are we going to do? And what am I going to present very quickly on here? Our approach is to go and get some pig legs and go and isolate the articular chondrocytes. We dedifferentiate them. In other words, we take them and make them execute a different genetic program so that they start multiplying. And then we try to redifferentiate them, giving them growth factors and other stimuli, and study this process by sampling what's happening at various time points from the starting material all the way through the process of going towards cartilage. We're sampling the cells. Now, we picked these 2 stimuli to start with, a growth factor TGF beta, and another chemical called vitamin C, because they are known to drive cells to different, or chondrocytes to different phenotypic outcomes. One of the key differentiators is the production of collagen 2, and here we've just verified what you're seeing is a stained cells where we're looking specifically for collagen 2 production, and as expected, the growth factor produces a lot of collagen 2, the vitamin C does not. So just validating some of the work. We are, as Danielle said, characterizing the behavior of these cells through gene expression. And so one way to just to preview some of the data that we'll look at to characterize that is to say, here's a gene. It may be expressed at a basal level or represented as a white box in terms of abundance. And here, a transcription, there's a stimulus added. The transcription factor binds and causes a change in expression, in this case, down. We started with 3, we went to 1 copy, and we'll represent with blue. We can do the same with upregulation. We'll represent upregulation with yellow boxes. And that means then we can have a panel of gene expression that's represented like this. So we're with that one gene, stimulus 1 turns the gene expression down, and stimulus 2 turns it up. We can do that for all or more genes and develop fingerprints for cell state based on gene expression using this method. And then we can start looking at more and more genes. So if we use RNA sequencing, which is what we did here, to identify individual gene expression, we can start making graphs that look something like this, where we have in blue, or each row representing the change in expression of a particular gene. Here in columns, 3 columns, we have a triplicate replicates here. We see the expression in the native tissue, the processed tissue, and then for days after stimulus. And here we can see that something is happening at the end of the experiment where these genes are being upregulated post-stimulus. Now we can start looking at more genes because we sequenced every gene that was expressed in the genome and start finding patterns of expression. Here's some genes that are stimulated in conditions that include vitamin C, and that are not stimulated in conditions that include growth factor beta. I'll skip that for the sake of time. Here's another example where we have genes that are expressed only in the presence of growth factor beta at different concentrations, but not in the presence of vitamin C. And so we can start differentiating these phenotypes and link that to the stimulus that was given. There's also a little box here to show that even though all of these genes are stimulated by growth factor beta, there is a subset that behaves differently from the others in a different condition. And that tells us that something different about the control, the wiring network, there's something different about how these cells are wired than these up here. So that's a hint that we'll come back to in a second. We can start doing simple analyses of what's stimulated in which condition. Here's a quick learning. Transcription factor beta stimulates glycolysis, pentose phosphate, glycan biosynthesis, cholesterol. Vitamin C stimulates a variety of different types of metabolic activities, showing us that these 2 stimuli are stimulating metabolism in different ways, which is quite interesting. We can look at the production of extracellular matrix proteins and other proteins involved, and very quickly, blue arrows are growth factor arrows and red arrows are vitamin C arrows. If they're pointing down, that means vitamin C or growth factor down regulates, and if they're pointing up, that means it upregulates them. And we can see patterns now start to pull apart patterns of expression that are similar between the cofactor, the stimuli, and that where the stimuli drive different expression of these genes. One, we'll zoom in here, one on collagen 6. It's actually a minor component of the extracellular matrix that's associated with the osteoarthritic state. When we saw this, it was pretty surprising because we thought transcription factor beta, the literature should tell us that it's driving cells away from osteoarthritic states. And here's an indicator that in our experimental conditions, we're actually doing the opposite of what we thought we were doing. And this would only have been possible by taking this approach where we're looking at all the genes that are expressed at once. It wouldn't have been information we could have gotten by looking at 5 or 10 of the traditional markers. And so this tells us something practical about what it is that we're doing in the lab and how well we are doing at driving the cells to the state that we want to get to. Last quick example of the type of information that we're interested in. Transcription factor beta is not just sitting there by itself when we add it. The cells are really smart. They control the amount of transcription factor beta that's active. So you can have a lot of it there, but they try to control how much of it is actually doing something. And they do that by having a bunch of other proteins that bind to it. And so here we've been able to start looking at what is the cell doing in response to these different stimuli to control the actual activity of the growth factor, of the signals that we are feeding the cells, which can then feed back in our experiments and tell us how we might control the levels exogenously versus what's happening endogenously. All right, to wrap up, because I know we're going a little bit long here. So that's to give you an idea of this approach of starting with a way of characterizing the tissue in ways that are much more expansive than what's traditionally done, giving us new insight into the developmental process and now new areas to go and start, if you want, poking to change the outcome of these tissue engineering experiments. So what about the wiring diagram? Well, that's ongoing work here. And it's inspired by work that I did with some other colleagues many years ago now in microbes, where we tried to use these high throughput technologies for profiling gene expression and finding transcription factor binding sites in a simple organism. And here on the right, we just say that it's a complex process where we bring all sorts of different types of information into a computational framework, where we try to predict what that regulatory network looks like. And in this simple organism that only had 2,000 genes, this network here was allowed us to predict the behavior of about 80% of the genes with about 80% confidence. And so it was a really explanatory network that allowed then us to go in and make modifications that change the behavior of the cell in a predictable way. So we'd like to follow up on this now with our model, and we have our preliminary network here. It's a hairball. It's hard to interpret for cartilage development. So this is a network where each of the nodes is connected to transcription factors, and each of these nodes are genes that have significant changes in expression in our experiment. We can look at sub pieces of the network where you have a target gene here, one of these proteins that modifies the extracellular matrix, and start to understand what are the factors that regulate this gene. You can look at collagen 2, one of the main structural components of the extracellular matrix. It has a much more complicated regulatory structure that's imposed on it. So this is our first model of the control structure. And our next task now is to go and here's a submodel. I took a simple one for explanatory purposes. But this model predicts that this transcription factor, ERG, regulates this matrix metalloprotease 8. We don't know how, but it predicts that that's what would happen. And so our next task is to go and systematically test some of these interactions and understand whether they're happening in our experiment, and if so, when they're happening in our experiment. Thereby, kind of, if you remember the network from Eric Davidson in sea urchin, where we saw the development of it, not the whole thing was active at once, pieces of it were active at different times. That's what we need to do now with this. All right. Just for completeness, we didn't have much time to talk about it, but we're also looking at the cellular dimension. So on the left is data from an experiment where we're looking at mouse cell lines, trying to use a different type of cell line that people are also using to study cartilage and development of this disease. And we've done a similar experiment to try and understand how similar are the different cell lines and their behavior in development, and try to understand how model systems like these cells on the left actually recapitulate the real behavior of chondrocytes. And the second thing we've been up to here is to start using what's called single cell transcriptomics. So instead of getting RNA from a population of cells, we're getting RNA from single cells now in a population, and trying to see if there are different behaviors that are evolving as the experiment goes on in a subpopulation of cells. That is, are they starting to stratify as you might expect in a growing tissue? So we have some very early results there that maybe some other time we can share. So in conclusion, I think just like to say that our approach is now providing what we think is the first high-resolution systems level evaluation of what happens to chondrocytes as they go through this tissue engineering process. And we're learning a lot more about what's happening in that process that's been known before. And we're starting to establish the foundations for concrete and testable models that we can use to, instead of ad hoc experiments, use hypothesis-driven testing to see if we can build new and better tissues. And then I didn't present them other than to just say that we're starting to look at other cells and their subpopulations. So that's it.
Wow. So I'm actually excited and speechless because I love the work you guys are doing at UC Davis, Father and Son. The one good thing, there's a lot of good things I learned from this research. I've done a lot of research myself in the lab with your dad. But the way you explain it with the radio analogy is so clear. It's so clear and simple, and everybody will get the message and understand exactly what you're trying to achieve and what you're doing. And overall, how does what you're doing in this lab, ex vivo, how can it help, in vivo research moving forward, or even in vivo knee replacement and all this stuff. So this is amazing work. I'm always excited when individual, an institution or a lab is doing something very positive in terms of looking forward. It's not an immediate outcome right now. It's like projecting forward. How can the research you're doing right now is going to benefit over 500 million people in the world? So I mean, this is the take-home message for me personally, and I love the work you guys are doing, and thank you so much for this work. Now, I will open it up for open discussion where I give the opportunity for my audience to ask questions. But before I do that, please go to the comment section. If you have not already subscribed to our channel, you will see where I have a public show and podcast YouTube channel, and please click on it and subscribe to our channel. This podcast will be posted tomorrow in this channel. So if you came in late, you will go in there. As long as you subscribe, you're gonna watch this video in full length. So now I will open it up. And also one more thing, the resources, some of these resources that both the one I mentioned and perhaps Professor Facciotti, Marc Facciotti, and Dr. Daniel Facciotti mentioned will also be in a public website, which are also listed in the chat. Please check out our YouTube channel and also our website. So now I'll open it up to the audience, and if you have any comment or question, please unmute yourself. You can also be on video and post the question that you want to. And also, one more thing, if you don't want to be on video, post the question, you can leave your comments and questions in the chat as well, and I can read it to Professor Marc Facciotti.
I have a question. This is Dr. Amanambu.
Dr. George Amanambu, go ahead.
Yes, I was kind of wondering, because say, for instance, in the knee, there is a lubrication fluid, the synovial fluid. Why wouldn't that help to reduce the incidence of degeneration of the cartilage in the knee?
Is the question, just to clarify, is the question whether supplementing with added synovial fluid might be a treatment or some? a different
I
question.
was just wondering, you know, based on the design of that location, and also
Hmm.
usually I have arthritis, and whenever I go to my doctor, he will give me an injection, but that might be for a temporal time. But I was thinking that the synovial fluid probably is supposed to help to reduce
Yeah. Yeah, if I can maybe understand, do you know exactly what he's injecting you with?
I never bothered to ask him exactly what he
Yeah,
was.
because I think I got that too, because I have also a busted knee, but it's hyaluronic acid. That's what they inject. You have seen maybe in one of the slides, it's one of the component of the extracellular matrix.
Okay.
And it is believed that, well, first of all, with an injection, you increase again the content, the liquid content. And that, it's like, how would I say it? A little bit of hydraulic kind
Okay.
of, you know, shock, like in a car, you know, you have that. But on top of that, the hyaluronate is believed not to be produced very well in arthritic knees, and therefore you supplement the knee with hyaluronate.
Very good. Okay.
If it is synovial liquid, and that one I haven't heard yet, that type of treatment, but in the synovium, there are a certain number of cells that it is believed, I don't know exactly how it has been demonstrated, but it is believed that there are mesenchymal cells in the synovial tissue that go and try to repair the knee. So, but I think most often it's hyaluronate that is injected. I got myself 3 injections, and it helps for a couple of months, 2 or 3 months, and then you have to renew the injection.
Exactly. Thank you very much for the insight.
Okay, I think, I don't know, Mark, will you add anything to this question or no?
No, I think that's it. Well
Okay.
described. Yep.
Dr. Wu, go ahead. I think you have your hands up.
Yes, can you hear me?
Yes.
Yeah, we can hear you.
Thank you, Dr. Mark and Dr. Daniel. This is survey research is wonderful, and is going to help a lot of people. I'm a registered nurse for many years. I work in critical care. I directly do not deal with people that have knee surgery or cartilage surgery, but I know that a lot of people will need cartilage here in America. My question is, how long will this research take to know the result, and how would it be tested on people to know the efficacy of the research? And was it done in animal first before? How do you all know to do this? Was it experimented in an animal?
Yeah, I'll take a stab at this first, and then Daniel can weigh in if he's got a slightly different view, because I think you've asked us to predict the future.
Speed up.
So,
she asked you to speed up the process.
Speed, exactly.
I'm not asking that, Andre.
No, so you know, anytime you try to predict the future, you're going to be wrong, right? So, but I'll give some estimate. I think I think we are many years away from having a perfect replacement. My hope is that we are within 5 to 10 years or so from making progress in improving the systems that we have now. So if we can find ways of improving the quality of the autologous implants that folks are making now, so that they last longer, they have a better quality, that you can imagine getting done in the lab in the next 5 to 10 years, and then you're looking at animal testing and then finally human testing to look at how well that works. So the timeline, the horizon is not tomorrow, but we could be overly pessimistic, and And sometimes luck favors the prepared, and you find something very earth-shattering next month, and that accelerates things.
Actually,
that's
in that
my
note, let me follow up with what Dr. Susan Oku asked. Professor Facciotti, currently it's UC Davis, your lab, partnering with any other institution in the United States to actually see where they are in this same in this type of research, whether any other institution are doing it, or is UC Davis, your lab, the only institution doing this research right now in the United States?
So there are many labs throughout the United States and around the world working on osteoarthritis, articular cartilage repair, so on and so forth. We are most tightly associated with Kyriakos Athanasiou, who used to be here at Davis and is now at UC Irvine. We've also worked a little bit with some folks over in our veterinary school here who are working on articular cartilage as well. But for the most part, the work that we're doing that I described today is happening in our lab. I'll add the graduate student, Hansu Kim, worked with some folks in orthopedics over at our medical school who are also interested in articular cartilage to start developing some of the experiments that weren't described here today, but were driving some of the work with the model cell lines that we talked about, the very briefly, the ATDC5 cells. But most of the work is described here was done in our lab.
Okay, thank you for answering the question.
Thank you.
Any other person that have any comment or questions?
Yes, in Dikaku.
Hello.
Hi, it's William Power.
Hi, William.
I want to say to Dr. Daniel and Professor Marc, from my personal experience and one of the things that I do and had personal experience with in this topic is I'll say that you're from my experience from a natural organic view of how the body functions to the way you're viewing this with your model and moving forward with I would say like a scientific way of working with the body, with the radio idea. Fabulous. You're on the right track in this way. It's a scientific way of looking at the hardware and all the things that you're doing. Just from my experience, and the reason why I'm endorsed by Harvard Medical School doctors, Oxford, and many others, including an Olympic athlete 14 years ago that was ready to retire at the age of 22 because they had a reoccurring injury, and they were told to retire at that age. And then they found out about me, and within 2 sessions, the issue, the reoccurring injury, and it had to do with tissue, the body fixed itself. And then they went on to the London Olympics and went to the finals. I personally have experienced 25 years ago a torn ACL and a meniscus and patella issue that I had surgery set up and worked with the surgeon in the way of my attitude was I'm going to do what I know, and I can always have the surgery. And the surgeon was a very cool surgeon that said, yeah, go about that. We'll see each other every 3 weeks. So the next time I saw the surgeon, I walked in normal. And he said, what are you doing? What the heck are you doing? I said, well, I'm working with the software, and I'm allowing the body to heal itself. And so I never, by the way, I never did have the surgery. And to this day, I don't even remember what knee it was. because they're both the same. And so I'm just coming from a place of no limitation. I'm coming from a place of viewing the body as an amazing machine that it's the software. It's as you put it, with the cell and the genes, and it's not the genes, it's the behavior behind the genes. So when you said that, I'm going, well, they're on the right track, because it's the behavior behind the genes that change the genes. The genes don't do it themselves. And working with tissue engineering and those other thoughts, I'm saying, if you continue in the way you are, in the manner you are, I truly believe you will have a magical breakthrough to realize, with this radio. I like that a lot, that little radio idea of having the right frequency, having the right signal, and working with it. And however else you work with the hardware or substances or injections, which personally I don't believe in, but it does work, to a certain degree. Having a combination of the natural organic way of the body, and the reason why the body's injuring is because of the issues that are going on. So I will say that the radio idea will help you come to a solution, a laser-like solution, which I've been able to find with my clients, and then they don't have to have these surgeries. The body fixes itself. I can't explain it. I don't come from a scientific mind. And
I think you're touching on you're touching on a really important topic here, which I think the goal of our work should really be to have as small of intervention as possible, and try to have the body repair itself whenever possible, and really to find what are the smallest interventions that we can make that help the body down towards the path of healing, without causing more trauma along the way, right? Ideally, what we would find is some simple. It may be an injection that helps the cells reawaken their ability to heal themselves, because in the knee they really have kind of locked themselves into a state where they can't do that once they've been matured. Without having to go through, all the complex stuff of making new tissue and that kind of thing. I think that would be the ultimate outcome. Is really
there could be something even more simpler than that, which if you I offer, if you ever get to that point that you want to think outside the box, to have that even more simple, and I can explain to you a bit more what I do or what I think of situations. Sure. Then it could be even more simple than that, to allow the body to heal itself.
That's great. Okay. So thank you, William, for the comment. It wasn't really a question. It was a compliment, a lot of compliment in different aspect of view, scientific or, naturopathic way of doing things. But like I said, immediately, Dr. Marc mentioned the idea model, simple model of radio. My whole brain was like going in different networks and stuff. So I love the idea. So continue using that model because it makes it simpler for people to understand the whole molecular analysis and pathways you're working with. Udoka has her hands up. Go ahead, Udoka, unmute yourself and pose your question or comment. I know we're a little bit over time, but let me see. Go ahead quickly.
Hi, my name is Udoka. I'm a student at University of Nevada, Reno, or just a recently graduated student. And
Congratulations.
Thank you. I was really fascinated by your research. I'm not very well versed on some of these topics, but I did do research with scoliosis. It's a little different than cartilage, but with scoliosis during my research hours at my university. And I was a nutrition major and a biology major. So I was going to ask about the TGFB1 and vitamin C and how those interact. I was just wondering if you had ever looked into, I have two questions, one after this, sorry. I was wondering if you ever looked into using vitamin A and seeing that relationship between those modulators, because I think vitamin A, like I read a research paper that it stimulates it kind of more at the mucosal lining. So I just kind of wanted to know kind of like the nutritional considerations when doing this type of research. And my second question is, are you accepting any students to your lab?
So the first question, the answer is simple, which is no. We haven't looked at vitamin A. I don't know if Daniel might have perused the literature more to see if there was anything there. I will.
Not that I found, Mark.
Yeah, I haven't either. But I will say, as we're getting started here, the selection of those two factors was deliberate, in that if you recall the cartilage having different zones of tissue, the TGF beta is associated with the formation of kind of the upper zones and the cell behavior that's consistent with kind of what's happening in those upper zones. And the vitamin C tends to stimulate the cells to have behavior that's more like what you see in the lower zones. And so we chose those two factors to start with to really see quite different outcomes of what might happen during development. I didn't have time to explain it here, but we've also looked at how they interact together. And then I think what you're bringing up is really really on point. That these are two factors of many that the cells are getting, that we need to do very similar type of studies to try and understand how the other factors, like some of the bone morphogenic proteins, potentially vitamin A, are impacting the development of kind of the genetic program of cartilage formation. So I think you're spot on in the sense that we need to look at other factors. We haven't looked at vitamin A. Am I accepting new students? The answer is maybe. Everything depends on resources. I would urge you to reach out to me. I can let me put my email in anybody can reach out here in the chat.
Perfect.
In general, I will say, graduate students to get into any lab typically need to be, apply to a program at UC Davis and get accepted into the program. And then after that, they start identifying potential research mentors. As an advertisement for our campus, I'll say that we have a slightly unique graduate program, which are split up into graduate groups. So you're not associated with a department, and faculty can belong to different groups. So you can go into a program that fits your interests in terms of coursework and that kind of thing, but still have access to a bunch of other faculty that may not be in, the department that you think might be what you're best fit for. And I can contact me. I can tell you all sorts of things about the programs here, and we can have a conversation about that.
Okay, perfect. Thank you. I'm applying to like special master's programs for pre-med and post-bacs. And I know that UC Davis does the post-bac, but if they were able to do a graduate program of things that I'm interested in, that's kind of framed like a special master program, I'd be very, but I will contact you further.
Contact me. Let's have an independent conversation about it, and we can get into more detail. Yeah.
Do you see that we are linking people with good networking right here now? So that's what we do. We're here to make sure that people understand the current situation of research, epidemiology, public health, medical, and in this case, scholarly research. So there's always rich information in scholarly research. I do not know if any other person has any other question or comments. I do not see any other comments. So that being said, thank you so much, Professor Marc Facciotti. Thank you, Dr. Daniel Facciotti, for accepting my invitation to feature in Public Podcast Show. This podcast show will be posted on YouTube tomorrow. Please again, subscribe if you have not already done that. The only way you support what we do here and spread the message is by actually subscribing to our channel and sharing this information to all your family and friends. Arthritis is a global pandemic. I said it. Yes, I said it. You know, I'm sure World Health Organization will say the same thing. But thank you for all the research you do. And without further ado, I don't know, Professor Facciotti, Daniel, if you have any closing remark.
I would just like to offer my thanks for people taking time out of their Saturday to hear what we had to share and for your questions. It's very much appreciated. And Ndukaku, thank you for the invitation. Fantastic.
Okay.
Thanks for same thing.
So
thank you all.
Right. And there's another opportunity. Marc mentioned that there might be future things coming up. So I will keep in touch. If there's any future improvement, I think this is the first time I don't know if this is the first time you're sharing this information, but anything coming up, we will always be in great contact, and you're always welcome. This is your home. You can come at any time. I contact Daniel or you to come here and present your study. Thank you so much. I'm humbly thank you for all the great work you're doing at University of California, Davis, and your dad. Thank you, Daniel, which is also my mentor. Thank you so much. And I'll keep in touch with you guys.
Okay. See you soon, Andy.
Okay.
Thank you very much.
Care.
Bye-bye. Thank you.
Thank you.
Father and son.
Thank you.